You're right, Rios. Get off stage.
Awesome. Okay, great. Well, let's get this kicked off. Welcome, everybody. I'm Brian Majerski, founding steward at Cerebrum DAO, and we're here with another episode of our Meet the Scientist series. We're hoping to bring this every two to 4 weeks to you, and we're hoping to feature a lot of our projects, but also feature a number of interesting things that we come across and scientists and research projects that we come across, even if Cerebrum can't get to a funding event. We still find a number of these things very interesting and deserving of a platform. And we want to just bring the science out to our community with these relatively informal chats with our scientists leading the important research that's leading toward fulfilling our mission. So we're here today with Dr. Luis Rios, and he's the principal investigator leading our FissionBio project. And I am happy to announce also that we did have earlier this year a successful crowdfunding for the project from Milestone 2, which we were anticipating we were going to go proceed to relatively quickly. And that was under the MITY token. So it's MITY, which is now listed on Ethereum on Uniswap. So you can actually buy this token and gain some governance rights in the project if it's of interest to you now. We got a lot of ground to cover today, and I want to get it rolling because there's so much to understand about mitochondrial dysfunction, and it is one of the core pillars of our hypotheses founding Cerebrum as we treat brain diseases and the pursuit of preventing brain diseases as a function of aging. Mitochondrial dysfunction is one of our core hypotheses that we wanted to fund a number of shots on goal in, and this just happens to be a very compelling project in that arena. Dr. Rios, and I'm going to call you Louis because we've been spending a lot of time together for the past couple of years.
Yeah, please do.
Yeah, well, we met back at the Lifespan conference in 2023
yes,
in New
correct.
York.
I was actually investing in the space under Vine Ventures. I was a partner there, so we were kind of just met as colleagues, both interested in the longevity space.
Yeah, no, it was fascinating, and I remember you had this research project underway at the time, and from the time where we met in 2023 until when we got reintroduced through the DeSci ecosystem, you had a pretty major breakthrough in this. So we'll get to that, but I'm wondering if you can start with just giving us a sense of who you are and your background and what led you to this category of research.
Yeah, absolutely. Well, thanks so much for having me and for taking the lead to promote projects like these. Yeah, so my name's Luis Rios. I have a PhD in chemical and systems biology, which is a little bit of everything that you need to do drug discovery. It's one part computation, one part biology, one part chemistry, and at Stanford, I did my PhD work on this project. So I was studying mitochondria and the connection between inflammation and mitochondrial dysfunction. And that's really the key part of my background for this discussion is I just became enthralled with this connection. And the big question was, how do we prevent mitochondrial dysfunction? So that's really consumed pretty much my whole PhD at Stanford, and then since then, spinning out Fission, initially Fission Pharma, but that project was like in the valley of death. So it's like so much academic research. There's this really interesting, promising science that comes up, and there's just a lack of funding to bridge the gap between the academic level and the industry level. And that's where Cerebrum enters my story, which is you guys came in and really are that bridge that takes something from the academic level and can help form a company and push the science to the point where I can get venture funding.
Yeah, no, that's one of the things I am super excited about with this project. It does feel like this is a very much a potential case study in why BioDAOs exist and what is out there that has true potential that otherwise would not get the necessary funding to bridge its way to the clinic. I'm definitely eager to get your thoughts on your experience in working with the DAO process and that at a later in the discussion. But I want to stay with the mitochondria right now. And so, what makes mitochondria an interesting therapeutic target for? I know we're talking about Alzheimer's and some brain diseases here, but in general, for disease targets.
Yeah, so the mitochondria is a large chunk of the cell. We're talking could be 10 to 30% of the cell is actually mitochondria, and in the case of neurons, it's more on the high side. Maybe a third of the a third of the cell is mitochondria, and mitochondria are particularly important for neurons because they're highly dependent on this organelle that does oxidative phosphorylation. This is like the process by which a vast majority of our energy is produced. So over 95% of our energy is produced in these little furnaces. And neurons, they are really at their metabolic limit. About 25% of all of our energy is burned in our brain, which means that there's a lot of oxidative stress. It's essentially a furnace. It's amazing that our brain doesn't cook itself. So there's this push to maximize energy output in the brain, but also to not oxidize everything around it, running these furnaces. So neurons are where you see mitochondrial dysfunction first, because they are the most dependent. So if you look at mitochondrial genetic disorders, they're often see neuropathy. Or if someone ingests a mitochondrial toxin, one of the first things you see is neuropathy. And that's because these neurons are so jam-packed full of these mitochondria, and they're so dependent upon them for their function. And also, other tissues as well, like muscle, are highly dependent on mitochondria. Other tissues in the body may not be burning as much fuel, and they may not be as reliant. So I think in the case of the brain, mitochondria really come into focus, and we see a lot of interesting signals from many different avenues of research that mitochondria are a big part of the neurodegeneration story, both in aging and also like early onset genetic disorders as well.
Yeah, no, thanks. That's a great description of it, and in particular tied to the brain and the energy consumption of our brain. These supercomputers that are on top of our shoulders here. So it's interesting because it seems like there's a lot of interest, certainly in mitochondria dysfunction for some time. Why do you think there's been so little success so far? Not many approved drugs yet for it, or targeting this.
Yeah, that's a great question. Yeah, why don't we have more treatments that specifically target mitochondria?
Mhm.
Well, I mean, for a long time, people were following the path of the antioxidant hypothesis that we could, try to take large amounts of of different powerful antioxidants like mito-Q or even like vitamin C, alpha lipoic acid, and that whole avenue of research didn't really pan out to produce any impactful therapeutics. And I think that's highly related to mitochondria are one of the primary sources of oxidative stress, and so I think the earliest kind of approaches were looking at antioxidants, but unfortunately that didn't really pan out. There are quite a few other targets that are mitochondrial in nature. I mean, because it's a huge chunk of the cell, thousands of proteins in the mitochondria. and they're just so completely interconnected. A lot of therapeutics, ultimately will affect mitochondrial function. They kind of have to in order to have any sort of, beneficial effect on the cell. But yeah, as far as something like what we're studying here, which is, one of the primary regulators of mitochondrial shape, something that's extremely specific to mitochondrial dysfunction. I think we'll see a lot more. It's really, I think, come into focus, like a lot more in the last, I don't know, maybe 10, 20 years. And it just takes a long time to translate that into effective medicines. I think the average time is about, 20 to 30 years from the scientific discoveries to when we actually see medicines in patients. So I think we'll see a large wave of mitochondrial therapeutics, either direct mitochondrial transplantations. It could be genetic, CRISPR-mediated, genetic diseases that are corrected. There could be a huge number of approaches that are targeted towards mitochondria that we're seeing in development. So I think it's really just a lag. Like it takes a while for this stuff to translate.
Got it. No, that makes a lot of sense. And I know one of the other things that I'm, really excited about for this project is there's some real innovation going on in terms of how to move things forward faster, which we'll touch on a little bit later in our discussion. But there are so many cool things that we're doing here with the Fission project. I love it. So let's keep rolling on this. So can you describe, so the project's fission, we're talking about mitochondrial fission. Can you describe in simple terms what is fission as it relates to our biology and in particular the mitochondrial fission part of it, and how does that tie to inflammaging or immunoaging and inflammation?
Yeah, I recommend that everyone at some point, just go onto YouTube and look up a video of mitochondria in action. You can use the search term mitochondrial dynamics, but basically mitochondria are not these static kidney beans that you see in the textbooks. They're extremely dynamic organelles that are constantly breaking apart and fusing together. So you see really this complex network of mitochondria that are all interacting, and the process of them splitting apart, fusing together, or being degraded is collectively known as mitochondrial dynamics. And this is a highly regulated process that is really at the core of many different parts of biology, between how cells divide, how stem cell identity, to inflammatory responses. So in the context of this project, really I was studying the connection between inflammatory signals, like stress responses, and how the mitochondrial network responds. And how it responds is the mitochondria undergo rapid fission. They break apart, they become fragmented. And this has a lot of really important impacts on the cell. The cell enters this sort of, some would call it like a cancer metabolism, like a state, a Warburg effect, where there is enough energy to use mitochondria, but the cell chooses to use more glycolysis, and the mitochondria become repurposed. They become broken into these small spheres that produce a lot of reactive oxygen species. And this is a part of this innate immune response that all of our cells have the capacity to undergo. So we see this stress response in neurons and skin cells, all over the body, and it's a highly conserved stress response where the cell deprioritizes energy production and starts to produce a lot of reactive oxygen species, which could it signals to other cells that there's a problem. If the cell becomes too overloaded, it's part of the way that the cell will undergo cell death, is that if this process goes too far, there's too much fission, the cell will die because it's not producing a lot of energy and it's producing a lot of reactive oxygen species. And so in the context of this project, that was the process, this innate immune response that's happening in the cells throughout our body, where the fission process becomes extremely activated. So big picture here, this is kind of the concept of mitochondrial dynamics, mitochondria are fusing, fizzing, and in the context of this project, the fact that we see rapid and extreme amounts of mitochondrial fission during the innate immune response, during the inflammatory response, was interesting. It provided us another avenue to treat inflammation because a lot of the other medicines were focused on the immune response side, right? So we have all these signals coming from outside of the cell, things like cytokines and these danger-associated molecular patterns of all sorts are coming from the outside of the cell. And we could block the immune system, right? We could throw a wrench somewhere in the immune response. Or we could look at the downstream, which is what's happening to all of our cells in the inflammatory state. And I think that's why this project is an interesting approach. It's coming from the bottom up. It's looking, okay, let's not focus on inhibiting the immune system. Let's look at what's happening to the cell during the stress response. And what you see, if you just look at an image of a cell, you see that the mitochondria and the mitochondrial network completely transforms within, you can watch it within 5-10 minutes. You can watch the mitochondrial network completely transform and the metabolic state of the cell rapidly shift. And so this is extremely problematic for neurons, which are so reliant on these mitochondria that when they undergo this stress response and the mitochondria become chopped up, the neurons die. And that's what we see in neurodegeneration.
Right, so that's the sustain part about it. So I guess from a standpoint of as you age, like there could be discrete stress, and stress signals that cause some temporary effects, but as we get older and older, that becomes more chronic, and then that continues to drive more and more mitochondrial dysfunction, which then started at the brain with neurodegeneration and cell death and neuron death to begin with.
Yeah, we see positive feedback loops. Mitochondria also have their own DNA, right? They're these endosymbionts that have a bacteria that our cells absorb, really, really early in evolution that created eukaryotes. And so they actually have a bacterial kind of genome, a separate genome. And the process of fusion and fission is actually the way in which this genome is spread throughout the mitochondrial network. As we age, we see the accumulation of DNA damage in the mitochondrial genome, similar to how we see the accumulation in our somatic cell or their nuclear genome. And that makes mitochondria much more susceptible to dysfunction as we age.
Got it.
And
so, go ahead. Yeah.
I was going to say that mitochondrial fission is because it's the way in which, not to get too deep in the science here. I know we wanted to keep this conversational, but I think this is important for aging, which is that when the cells are undergoing a lot of fission, they can't exchange DNA. And this is how the mitochondrial network purifies itself of damaged DNA, is that the good mitochondria find the right DNA, and the mitochondria with damaged DNA, they get selectively degraded. And so you see through this exchange process, a sort of evolutionary sort of genetic process, we see that if we get good exchange of DNA, that's our way of getting rid of damaged mitochondrial DNA. And if you get too much fission for too long of a period of time, you don't get good DNA exchange throughout the network, and you end up with an overall network of mitochondria with a lot more damaged DNA.
Got it. Amazing. So very much this is an upstream intervention. Like we're targeting certain disease areas here, but fundamentally we're talking about improving mitochondrial function, which will then lead to less inflammation and less potential susceptibility to a variety of chronic illnesses.
Yeah, and inflammation is a tricky one. It's a double-edged sword, right? We need an inflammatory response for so many reasons. Without it, we would all die of infection very rapidly. So the big question is, how do we have powerful anti-inflammatories, a process that we know is linked to many diseases? How do we generate more powerful anti-inflammatories without suppressing the immune system? That's the double-edged sword of the anti-inflammatory strategy. And I'm hoping that focusing on the metabolism, the metabolic health of the cell, instead of focusing on the immune system itself, will allow us to generate a more powerful anti-inflammatory effect, because we're not directly poisoning the immune system. We're not blocking cytokines or any of these other signals that are essential to our body's ability to detect and defend ourselves from infections and so many other processes. And that's the key part was that this DRP1, this fission response, the key insight, I mean, people wanted to drug this target for a long time. The key insight was that there was a specific protein that was only involved in this inflammatory response. And by getting incredibly precise about what part of this system we were blocking, we were able to find a way to block this inflammatory metabolic response without blocking the normal everyday function of mitochondria.
Yeah, that's the amazing potential breakthrough here, as we are really talking about addressing immunoaging without suppressing the immune system itself. I think we've seen this in, I don't know if I've got this exactly correct, but I think there was a whole category on Alzheimer's that was targeting the amyloid, and it was the gamma secretase inhibitors. And so like they wipe out amyloid, and amyloid's actually a necessary inflammatory response in the brain. And so obviously you don't want too much of it, but you need some of it. Otherwise, you're not going to end up in a good outcome. So that's very, very interesting. So you want? Can you talk a little bit more about the therapeutic target? I know we got to be careful with intellectual property, but this DRP1 Fis1 target, in layman's terms, maybe a little bit more about why that's important. I know you hit on a good amount of it just there, but if there's anything else you want to just crystallize there before we move on.
So DRP1, just very broadly here, because we can go down the rabbit hole really far on this topic. But it's a protein, an essential protein for the regulation of mitochondrial function. It's basically a lasso that cuts the mitochondria physically in half. And it was a target that people wanted to drug and have been drugging for a while. They've been studying it for 20 years at least. And we know that it's linked to neurodegeneration and many diseases. But the problem is that it has essential functions. Like so many proteins, the body probably wouldn't have it if it wasn't doing something important. And so the only way we really knew how to drug it was through its active site, which is non-specific. It just shuts off DRP1 generally. And we know that if you shut down DRP1 generally, you can actually induce inflammation, because it's like the gardener of the mitochondrial network. It's the scissors that our cells use to clip off damaged parts of the mitochondrial network. a process known as mitophagy. So drugging this target directly, while clearly linked to disease, we couldn't do it because I don't think it's a good strategy to target it. There's a lot of studies on this, genetic studies, where even if you knock it down by just a little bit, you can, the cells that will actually end up having neurodegeneration anyway. You kind of have the same response uh, because it's so essential for maintaining mitochondrial function. And the key insight of this project came from the discovery of a peptide called P110 about 14, 15 years ago at Stanford University. And we didn't really know how this peptide, where it bound on DRP1, but we knew that it selectively blocked the interaction between DRP1 and one specific protein, FIS1. And so DRP1 was free to do, all of its interact with all of its other binding partners, of which there are probably dozens of other essential proteins that it interacts with to do other important functions. There was just this, it just blocked the interaction with this one protein. And we didn't know how exactly it did that. We just knew that it worked. We knew that it bound to DRP1 and blocked this protein-protein interaction. And so that was the key insight there, was the discovery of that peptide. And then this peptide went on, 13 different disease models, just dozens of interesting papers with this peptide, really promising. A company was actually formed around it, Mitoconics. But the company couldn't raise because people fundamentally didn't believe that a peptide could be a medicine. It was limited. It can't be a pill that you take every day. It would have to be infused IV, basically continuously, because it has the peptide can get chopped up by proteases. It's not. People didn't believe a peptide could be a drug. That's the ethos there is changing, but fundamentally there were problems with P110. While an amazing proof of concept that prevented it from entering the clinic. And that's where I came into the story. Looking at this, I asked the question, perhaps we could find a small molecule that mimics the activity of this super promising peptide therapeutic that has efficacy in all of these different disease models, from neurodegeneration to sepsis to ischemia reperfusion injury in the heart to IBS. This peptide had all of these really interesting efficacy endpoints in animal models. And, really the key unlock was, all right, if we can find a small molecule that mimics this peptide, has the same activity, we'll be able to turn that into a drug. We know very well how to turn small molecules into drugs. This is a well-trodden path. And the technology around peptides is newer. A lot of it's still in development. A lot of really good, interesting work happening there. But small molecules was the tried and true path. And I got lucky. I used in silico modeling and screening to actually discover that small molecule. It's called SC9, and that was published in 2023 in Nature Communications. So, as far as IP, we can all talk openly about. This is public knowledge. This research has been around for a long time, with P110 and now SC9. And so, where are we going to go from there? I guess is the big question. I don't want to ramble too long here, Brian. You take the reins. I'll ramble on this one for an hour, easy.
No, you're doing a great job. This is tying and weaving together exactly where I was hoping this was going to go. So that is fantastic. So you've kind of taken the small molecule approach, and you've actually found an interesting pathway. So far, there hasn't been any success on this. What do you think stands out for what led you to this promising project right now that you've done differently that the industry hasn't done?
Like this project. What have we done on this project that industry hasn't done? Well, really, it's just this allosteric site, this new binding site that I just discovered. How we actually find these drugs and develop them is complex. It's a protein-protein interaction, which is trickier to drug than like an active site, something like an enzyme that or a receptor that evolved to bind to a small molecule. The shape of these binding sites is different, so it's actually a much trickier problem to find these protein-protein interaction inhibitors on these allosteric sites. Uh, you know, and the system itself. How do you study mitochondrial dynamics? How do you set up the right series of assays to know that you found a compound with the right effects? That is also a very niche field. This drug discovery within mitochondrial dynamics. There's a lot of pitfalls in this system. Everything's kind of connected to everything with mitochondria. We're talking maybe like a third of the cell. Pretty much everything that the cell does somehow involves mitochondria on some level. Like, at least it's closely related. So it's a tricky system to study, and it's a tricky system to find drugs in. And I think I was lucky to make this discovery and to be in one of the world's leading labs, the Daria Moshley Rosen lab at Stanford University. Has been studying the system for a long time, and so this knowledge, this wisdom, was passed down. And I think that's giving us an advantage. Is in research, you get caught up on so many unexpected little snags. There's a million points of failure, and so this sort of research wisdom that's passed down is essential to the success of a program.
No, indeed. So let's turn our attention to the project itself now. And maybe you can talk a little bit about the research you're doing right now, where we are. The Cerebrum DAO communities come together. The DAO itself has committed to fund this project, and then we did a IP token crowd sale to bring in people from outside the DAO to own a direct governance right in the project as well. So I think people are definitely would love to hear how it's going and what the roadmap looks like. So maybe talk a bit about the first milestone, your plans for that. Where are we on that? And then we can take it from there.
Yeah, so happy to report that our first milestone is complete. We the first step was to scale up the in silico screen. So as I mentioned before, I discovered SC9 with an in silico approach, which means we have a three-dimensional model of the protein, trying to predict, we modeled the binding site, we're trying to predict which ligands, which molecules will bind to that site. And this, the technology around in silico screening with AI, has now advanced rapidly. And so the first step was, all right, how do we make this in silico approach much better? Because the initial molecule that I discovered, SC9, while it was an amazing tool compound, a proof of concept, does not have drug-like properties. And so the first task was, can we design in silico a molecule that can bind to this binding site and also have drug-like properties? And the search space here is astronomical, right? So we're looking through this vast chemical search space, something like 10 to the 60 possible drug-like molecules that you have to consider, right? So now with AI and the just vast compute resources at Klein AI, this company that I also founded, we were able to do a billion compound scale screen for incredibly cheap. So at the DAO, we're trying to do things as cheap as possible, as fast as possible, trying to make drug discovery more efficient. And we're lucky that we have this new technology that allowed us to do what would have cost probably $500,000 before for, under $25,000 initially for the primary screen. And then, we used a secondary a part of the screen called molecular dynamics, right? The first part of the screen is static. The second part of the screen, you're actually playing out physics of how the molecules interact. And that's a much better predictor. So it was a huge effort to do this massive in silico screen. That was one of the key parts of first milestone, which was complete. We have a lot of really interesting hits that we're excited to screen. And then the assay development was the second part of that. So, as I mentioned, there's a lot of intricacies and pitfalls when it comes to screening for these types of drugs. And I had to transfer that technology at Wisdom to a CRO. So I had to work with them, and through quite a few iterations, we're able to get the assay working with enough sensitivity to screen for molecules. And they actually now have a test set of 50 molecules that they are screening as we speak. So we're actually beyond milestone 1. We're into milestone 2. And we haven't even cashed in on the milestone 2 funding yet. So we're ahead of schedule, and I think that's all thanks to really just the innovations around in silico modeling and AI, which we're going to leverage in order to make the development cycle as short and as cost efficient as possible.
That's amazing. So what then will be the next steps when we look at milestone 2? What's the roadmap for milestone 2? I want to dive into the AI in a minute, because like this whole concept of faster, better, cheaper. It's been a mantra here with Cerebrum, and I think with the BioDAOs, because we are dealing with limited resources in the nascent stage of this industry. And we need to prove that we can do something different. And doing something different is finding opportunities like this with people like you and researchers like you that otherwise would not get the funding to kind of bridge through the translation gap. And then part of being able to do that with the resources we have is to be able to do it better, faster, cheaper with the current technologies that are out there. Without shortchanging the science, obviously, right? So we can't do that. But better, faster, cheaper doesn't mean less scientifically rigorous or less evidence. And so that's why I hesitate to use that phrase sometimes, but because people question, then they're like, well, is it going to be scientifically legitimate? We're going to go through the normal process and channels, but I do think there's a way to speed things up, and I think we're proving it partially, or we're hoping to prove it with this project too.
Yeah. Better, faster, cheaper in the context of preclinical drug discovery is really how many molecules do you have to synthesize in order to find one that has the drug-like properties that you need. Traditionally, that has been in the thousands of molecules, something like on the order of 5,000 molecules you have to synthesize in order to find the right one. And that's with expert teams, armies of medicinal chemists and experienced drug discovery people, a massive pharmaceutical apparatus doing this research. And that's incredibly expensive.
So what does that look like in terms of time and cost on average basis?
Yeah, I mean, it varies widely, but we're talking in preclinical, just a preclinical phase, millions of dollars to synthesize all these molecules and do all the assays that need to be done in order to de-risk something sufficiently to bring it through IND-enabling studies and eventually into the clinic. So, the hope here and what we've seen from early, this technology is very early, so we can't, there's going to be some critics of it, but what we're seeing is that we don't have to make and screen as many molecules as before. we can make better predictions about which molecules to make next. So while we may have had to synthesize 5,000 before, and that's after you do a high throughput screen. So you may have synthesized and screened hundreds of thousands to millions of molecules first in a high throughput screen just to get to the step where you have to make the 5,000 molecules that are custom. And so we've completely eliminated the need to do a high throughput screen physically, hundreds of thousands of molecules often. Just you could imagine the amount of work that goes into making all of those. And we may have to make something on the order to 50 to 100 molecules in order to find, we're talking 99% efficiency gain in the amount of chemical synthesis we have to do and the amount of molecules we have to screen in the lab. It's really exciting technology that's been in development for a long time, and it's getting better and better with the amount of compute infrastructure we now have. In this screen, we were able to spin up tens of thousands of GPUs and CPUs, and leveraging this machine learning algorithms blended with biophysics. It's amazing what we can do now. And we're making predictions that actually we're seeing hit rates on the order of 10 to 20% on targets that have never been drugged. So it's very exciting for preclinical development, which is very important for a group like Cerebrum, where the risk is high, so the amount of time and funding has to be minimized when it comes to early stage preclinical projects. And I think technologies like AI and machine learning are going to allow us to run a lot more experiments, to roll the dice a lot more times, so that we have a much better shot of finding the winners.
Yeah. So there are a number of players in this space of AI preclinical drug discovery. You mentioned your company, Klein, that we're working with. What distinguishes it? Is there something unique in the mitochondrial field, or is it just general on a platform basis versus some of the other players in the space? And what does that landscape look like today?
Yeah, there's a lot of really amazing computational teams that are springing up around AI and drug discovery. There is an effect where, because all of the upside, pretty much all of the upside, is in the asset itself, the drug that you would eventually market, all the talent and resources, or at least most of it, is being sucked into internal development pipelines. Right. So, like Google DeepMind, its technology was eventually pulled internal, and there's many other large, really interesting companies developing this tech. And so that actually left an opportunity, a company that could come in and see that, okay, maybe the upside is not in the tens of billions, but the upside could be in the hundreds of millions to even up to $1 billion, and basically develop the technology that's been tucked away inside of these AI drug discovery pharmaceutical companies and make it available to everyone, and do it with a team that's very lean. So Klein has been able to produce this software with a team of 4, which means the overhead cost at Klein is really low, right? It doesn't take much for us to be profitable. And I think that's really what distinguishes Klein. Like we have the tech that a lot of the big companies have produced, and we are making it available as cheap as possible to as many early stage labs. Academics haven't even been able to afford to run these massive cutting edge screens, and small biotech companies. So it felt like there was just the early stage companies were underserved by the in silico offerings. And that's what I experienced at Fission. I came out of grad school with this awesome proof of concept molecule ready to launch a company. and I'm looking at what it would cost to run an in silico screen, and it was in the hundreds of thousands of dollars. And for a project at that stage, like that's like most of your budget. Maybe you can raise $500,000 at first with like a proof of concept hit molecule. So there was this gap, right? There's a demand for a lean company that had cutting edge tech that could keep its costs down. And that actually is increasing the amount of in silico screening people are doing. So a lot of these early stage companies were just going without, this technology because they couldn't afford it. You would need a computational chemist. You would need software licenses that cost hundreds of thousands of dollars. And you would need compute infrastructure, someone who knew how to like spin up all of these GPUs and CPUs. And so it just made sense I basically created Client because I just couldn't, I found this problem in Fission. It just seemed
You
like,
solved
okay,
your own problem. That's a
yeah, I
classic
solved my own problem.
startup
I
case.
came in and I said, this doesn't make sense. This tech should be available to everyone. It should be cheap. You don't need a organization with hundreds of people to build the software anymore. And we have really just the most essential key people at Client. So we've kept our costs very low, and that's really what distinguishes us. We're not claiming we're so much better at Client than anyone else. We're just making it we're making the cutting edge tech available to everyone at
Incredible.
a price point of something like $10,000 to $50,000 instead of $500,000.
Awesome. It truly is amazing, and I think it's going to be very interesting to see where the field goes over the next several years. And clearly, we're going to be the beneficiaries of it too. So I feel like we're in the right place at the right time. So I think we left one piece hanging about milestone 2 on the Fission project, and so do you want to share like where are we heading right now? Like what are the next phases? What's the next milestone we're trying to hit? And then I can dive into one more question.
So we are ready for screening. So we're actually launching the first set of 50 compounds. We will screen those through a series of assays. Right? We have 3 different assays now up and running that we can use to screen these molecules. And now we're in the process of having these molecules synthesized and tested. So the hope is that at the end of milestone 2, we'll have a collection of hit molecules that have different scaffolds, right? Ideally, I'd like at least 3 different scaffolds, types, major types of molecules that are binding to this site that have the effect that we're looking for in cells and have drug-like properties, like properties where these molecules could potentially get into the brain. And so, that's what we're doing in milestone 2. It's just a screen.
Awesome. And what do you think? Like, what does that timeline look like, roughly?
So it takes about amazingly from providers like Enamine that can now offer trillions of molecules for $100 each, and you can get them in 2 to 4 weeks. So the first step, once the funding on milestone 2 comes through, we're going to send a good chunk of that to Enamine, and they're going to make a lot of these molecules that are top hits from the in silico screen. That could take about a month, and it may take us about a month to screen them through the 3 different assays. And you can always add a month in there for good measure of various delays that happen. A lot of unforeseen delays, just honestly bureaucratic logistical things that get in the way, contracts that need to be signed, or like it's funny that the science doesn't even consume all the time. It's like maybe half of it's bureaucratic logistical and half of it's science.
No, but the pace is really great. I mean, we're talking about some potential hit candidates in a matter of months, and I think, as DeSci is an emerging ecosystem, and we've been successful at generating some, buzz, some interest. We're pulling in some very talented people. People are looking to say, okay, well, what is this all for? Where are the results? Which
Yeah,
projects are performing? And, when are we going to see the promise of outcomes for, people, potential outcomes for people in the clinic? And I think,
yeah,
this pace and this level of innovation is really important for people to hear and see that, the stuff that these bio DAOs like Cerebrum DAO are funding are real, very innovative, and potentially massively impactful.
and a
And the results are
critical,
happening and real work's
yeah,
happening.
and a critical link in the ecosystem. So as we've seen a contraction in venture capital spending in biotech, right? We've kind of gone through this biotech winter of sorts. That means the funding is going later and later. VCs are looking for lower risk projects, projects that have, at the minimum, a good hit, more likely a lead molecule, which is a break in the chain, right? We have a lot of interesting tech coming out of academic labs, very, very small startups that need to bridge the valley of death. And, Cerebrum DAO's coming in and providing that bridge. It's absolutely essential. Hopefully, we see a shift in the venture space and more funding will come back into early stage projects, higher risk projects. But that's, I'm just really glad that the DeSci community has come in and filled this niche.
Yeah, and we're excited that we have a community that's willing to do this. And as we start activating, the community of people that are actually impacted by these illnesses that we're trying to solve for, see value in this, they can bring their time and talent and their money to the field, and that gives us much more ability to fund a lot more projects and even into later stages too. So DeSci could be a lot of that solution in addition to re-energizing some of the biotech, life sciences, venture capital space too. Cool. We have about 4 to 5 minutes left. I usually like to leave time for Q&A, so I will
Yeah,
open it up for Q&A. I do have a question for you to close with if we don't have questions, but I did want to give the audience a chance to kind of pop in with some questions. So if you have them, I think you can drop them in the comments, or we'll try to pull them off of X Spaces and into this broadcast room here.
also, yeah, feel free to reach out to me personally. I love chatting with people about the science. So if we don't get to your question right now, just reach out.
Awesome. How can
And
people reach you?
they can find me on LinkedIn, yeah, I think LinkedIn's the best place. You can give them my email too for Fission, and just send me an email. Happy to chat with anyone.
Awesome.
But yeah, let's see if we can get. I mean, if you have any
I'll see if we have
questions,
any more questions. We
get to.
covered a lot of ground here. And we'll see. Okay, we got one coming in. What do you think the future of AI and what do you think the future of AI is in drug discovery? Will there be more small companies like Klein?
Yeah, so I think one of the things that's really exciting about AI is that because it can be often built by smaller teams, and these companies can be run with because they can become highly automated, we're going to see small companies that are extremely efficient doing things that large companies used to do. And so companies like Climb, I hope there will be thousands of them. That's going to drive the price of everything down. Right? We need this deflationary effect in this extremely expensive ecosystem. And ultimately, that will lead to a lot more and a lot higher quality research. And so I think the future of AI and drug discovery, yeah, we're just at the very beginning of it. Fundamentally, we're limited by data. That's what's slowing everything down. So at Klein, there's an effort to generate one of the largest drug protein interaction databases ever built. And hopefully, it seems like data is really the key currency in the AI space, right? That's what differentiates the different companies. There's publicly available data, but there's proprietary data. We'd all be better off if we just all shared all of our data. We just threw it all together in one big pool and we built a super AI that had access to everything. But that's just not that's just not how it works.
Got it. I think we can spill over a couple minutes. We have one more question coming in here. So the question is, you can screen one target with billions of compounds. Can you effectively screen one compound with multiple targets of billions of compounds, or billions of compounds with multiple targets?
So Klein just ran a screen 7.2 billion compounds across 4 targets simultaneously. Like the scale and the capabilities are really tremendous. With generative modeling is particularly good at multi-parameter optimization. So you can, instead of going from the bottom up where you're considering molecules at random or intelligently selected molecules that are enumerated, the generative model comes from the other side. It kind of acts as a medicinal chemist of sorts, and it actually takes molecules and makes chemical modifications to them based on its training, looking at different chemical syntheses and different types of drug-like molecules. And so coming from the bottom of looking, using a generative approach, you can ask the generative model to iteratively optimize through as many parameters, right? So it'll score, it'll make a series of mutations, a series of new molecules. It'll score it across multiple parameters, and it'll move iteratively to find a global maximum. That's probably the most efficient approach, and that's one of the newest and most exciting approaches in chemical generative modeling.
Fascinating. Well, truly new frontiers.
Yeah.
So I'll do a closing question here. Going back to your roots on mitochondria, how do you see the field of mitochondrial targeted therapies going for over the next 5 to 10 years? What do you think the future is for this?
I think big picture, I really hope that mitochondrial transplantations work. It's a really interesting idea. We haven't been able to manufacture and get that to work. That would be amazing. It wouldn't address the problem of inflammation, but what do you do in the case where you already have a lot of damaged mitochondria? How do you go back? And one of the hopes is that we can just replace it, right? Like kind of like the ship of Theseus or an old car where you can replace parts as they break. People are hoping to do this with mitochondria. I really hope that works. We could hopefully wholesale replace them. Ultimately, we'll still have to block the damage to those new mitochondria you just put in, which is what we're hoping to do at Fission, is to cut the link between inflammation and mitochondria. But there are, there's a ton of different exciting therapies. We're out of time. I do have to jump as well. Yeah. But again, feel free to reach out to me. I'm happy to chat about this for way too long till your eyes glaze over.
Incredible. Well, thank you, Luis, for spending this time with us and for all the work that you're doing, pioneering and innovating in the AI screening space as well as in the mitochondrial space. We're really pumped about this project and working with you. So it's been a pleasure, and I know it'll continue. And thanks for spending time with our community today. And thank you all for attending. Thank you all for attending the Meet the Scientists series, and we'll be on with our next one in a couple of weeks.
All right. Bye.
Bye.